Welcome back to Frozen Bytes, the Frozen Ark podcast. If you missed any of our previous episodes where we've discussed the virtual ark, climate change, coral reefs, invertebrates and cryo-arks, you can find them on our YouTube channel. Today, myself and the other professional training year students at the Frozen Ark had the incredible opportunity to interview Ann Clarke. Ann is the last surviving founder of the Frozen Ark. She continues to move the charity forward with her inspirational enthusiasm and drive. We started by introducing ourselves to Ann and then asked her a few questions about her work and life. I'm Rhian, and I was living in Cardiff for the last year, doing a placement with MAF, and now I've come back home to Essex, where I'm just living and working remotely for a couple more months.
So you're a PhD student?
No, we're undergrad students.
You're undergrads doing biology, zoology. What year?
This is our third year.
My name is Cléa. I'm also a biology student at the University of Bath. And yes, we've done our placement this year with Frozen Ark, and it's been really good. Even though it's all been remote, we've still done a lot. And we got to go into the office and the lab in Cardiff like a couple times, which was nice. Right. And I've left Cardiff now, and I'm spending the rest of summer in London.
And what's your plan for the future?
Gonna finish my degree first and then take it from there, but no, I'm really interested in like conservation obviously, and also immunology, which I know
my other subject.
It's quite similar. But so either one of those two pathways I think I'd like to go down.
I'm Lydia. I do biochemistry at the University of Bath. I was also doing my placement in Cardiff, and I've been doing a project looking into making biobanking more like environmentally friendly, which has been really cool.
So what does that involve?
Yeah, it's sort of looking at every process and how can we make it a bit greener, a bit softer on the environment, and a lot of that is how you use freezers, even simple things like.
And collecting the
filling empty freezers with polystyrene boxes to make sure retain the air and then that kind of thing, and then reducing plastic where we can, and there's a lot that we can do.
That's sort of modern day storage almost for the long term, i.e. the DNA doesn't go off because the freezer went down by 2 degrees, this kind of thing. Mm-hmm. That's very important.
Yeah
Storage.
Definitely.
I'm Hollie. I'm one of the PTY students based at the University of Nottingham.
Oh, you're in Nottingham?
Yes.
Which department?
At Nottingham Trent University. That's where I study, but I'm doing my placement here at the Frozen Ark at the University of Nottingham.
All right. You're working in the lab at the moment.
Yes, I'm working on the particular snails that has been collected. So I'm putting them in a database, doing some DNA extractions and just doing some work with hangers here.
Hi, I'm Hollie. I study zoology at Nottingham Trent, also a placement student. I was originally meant to be based in the Nottingham office with Olu. However, just because of COVID and everything, I've just been working remotely from my home in Kent.
Right.
I've been kind of continuing the project from previous year of the Prioritisation tool. So just trying to help finish that off and finish the last little tweaks with the coding and that sort of stuff to try and get it all finished.
Oh, well, that's very interesting. Well, there's quite a number of us, aren't there? It's good. Yeah.
Okay then, so I think to start with, it'd be nice just to have like your version of the story of the Frozen Ark and how it all started. If you could just talk us through that sort of as you experienced it.
Well, yes. I mean, it's quite a long time ago now. I was sort of working it out. It's about 20 years since the idea first cropped up. And it was because of the snails. And so in 1960s, I think we were married then, but we might have been just engaged. My husband, Professor Brian Clarke, he was professor at Nottingham zoology at that stage. And I was actually working towards, I was working in the lab for some time, and I was working towards being a journalist post-school, but they told me I had to wait 6 months. So I thought, oh, great, I'll join in 6 months, have 6 months off. And my father said, what are you going to do now? So I applied. I thought he'll say you'll have to earn your living. So I thought, well, I won't. I better look in the paper. And there was a job to work in the Department of Zoology in Oxford as a technician. John Gurdon, who worked on Xenopus and has become really rather famous, and I was his technician for actually 3 years. And this converted me into a biologist. So then I took a degree. I met Brian and we got engaged, and he got a lectureship in Edinburgh. And so I thought, I've got to get on with this biology, so I better get a degree. So I applied and got a degree to do a zoology degree. And then I did that. And then the problem of the snails came up, and Brian collected snails for his PhD called sapir. And they're the snails you find in hedgerows of England. And he was doing his PhD. So this was in Oxford. And I used to go snailing with him. And then he and his PhD partner were talking together in the lab one day, and at the library. And they picked up a volume of Crampton. There's a man called Crampton who'd studied the Polynesian snails and all the varieties in the French Polynesian islands, Tahiti and Maria and things. And they thought, wow, that would be a wonderful thing, because these are snails with all different kinds of types and species, and the whole business of biodiversity, interspecies and variability were interesting tools, particularly at that time. And so they applied for grants to go to Polynesia. And so when Brian and I got married, first thing we did was set off for Polynesia, from one of 7 trips I think I went on. And this, of course, then involved the zoos and things when we came back with snails. So we got in with the zoo people in London Zoo and Natural History Museum people in London and in Scotland. And the whole thing was created in about 2020. And so we went to Polynesia and on a boat, and it took us 6 weeks. And we were on a boat called the Carry-Moon. And we weren't bored for a minute, because we kept catching. We had a net, we caught things in the sea. We had a small microscope, and we used to look at those all day. And it took us 6 weeks, and then we got there. And there was Jim Murray, his wife Biz, Elizabeth, me and Brian were the first 4. And we started to repeat. This book of Crampton showed us all the places where he'd collected snails from, and we just repeated his going to the same places in this most beautiful, unbelievably gorgeous island of Maria, which is just off the island of just a little bit away from Tahiti, but much smaller, and it has huge volcanic mountains and forests and these snails. So we started the snails, and that was all very interesting. And then we went 3 or 4 times, and all went well. And we gave them to the zoo and Natural History Museum, and all went well until terrible day about 10 years after that, 1960, about 1978 or something like that. I can't remember the date. The French introduced the edible snail, Achatina, to eat. And that was the one from South America. And unfortunately, no one had thought it was a big snail, and no one had thought that of what would happen. So they introduced it to the islands, and it had no natural predators, so it just multiplied up, and it started eating all the crops. And then they thought the French government, the French colony, Tahiti was at the time, and they said, we've got to get rid of these. So they introduced the cannibal snail, Euglandina, from South America to eat the Achatina, but they didn't do any experiments, and it didn't eat the Achatina, it ate the partridge instead. So the study of evolution, a scientific study, became a study of conservation. And we went back, we got the zoo involved, and Paul Piers Kelly and others came out and collected snails, and we built them up in the museums and the labs, and we brought them back to Nottingham, and they're still going in Nottingham, I think, yes. Quite a few of the different 68 or so species that exist. I think that's all over. I think there's about 12 in where we were collecting of the different types. Taeniata was the smallest, and Sucheralis was the biggest, and they were all in between. So we did that for more years and more years. And so this story of extinction, well, yes, the story of extinction and the story, and then London Zoo's got to taking them back into the wild and letting them go, and that project is still going. Very sorry to have lost the leader to COVID, so we'll have to do without him. Get someone else to go out to Polynesia and look after all the samples that they've released successfully, so that the whole species, group of species, isn't lost. So this is really rather a very early example of finding. It's an attempt at conservation of an animal that is endangered by mankind's unthought and disastrous things that we're doing, which is killing off species, and then we've got climate change now, which is added to the problem. So there's my story. Do ask me any questions.
I mean, it's an amazing story, and it's very lucky that you did have to wait 6 months before you got to start your journalism job. Otherwise, you wouldn't have been involved in it at all. So that's a good thing for the Frozen Ark, for sure. Obviously, so sad that it did become a story of conservation rather than what you set it out to be, but ended up having a good cause in the end, I suppose. And so what do you sort of think of as the importance of biobanking and things like that for endangered species conservation in general? What do you feel it can achieve in terms of helping these species which are endangered?
Well, I suppose you've got three or so levels. First of all, you've got the zoos. So, if we get the last elephant into his last half dozen elephants, it would have to be because of genetic reasons. Breeding and things, not too much inbreeding, and that is actually one of the problems with zoos, is that quite a lot of the mammalian species, and I don't think we've studied the others to the extent, but it's probably the same for all other groups, is that they get inbred, and then you have all the problems of inbreeding, and eventually lose the species. The zoos are important, but you mustn't let species die out in the wild, because that's when you get genetic variability comes into it. So I think conservation and look what's happening with climate change, that's a whole new story which is going to add to the problems we face. And I think is probably going to be essential. I think very recently, and I'm very interested to hear your latest ideas where I was hearing from Jude, is probably, it's so difficult to do all the vertebrates, say, but I think they'll manage the big ones, the lions and the tigers and the elephants and the ones that people, leopards, or, all the rest of it. We haven't even started at all about what's under the sea. But we have these quite good ways of preserving things, -80, -180, freezing, freeze drying, all these methods that enable us to at least keep the blueprint. So there is possibilities of introducing variability into an inbred population. And as things develop, we will get more into how you can basically recreate maybe a species. But you need the genetic codes of them all first to be able to do that. So I think not only is probably freezing down the DNA and cells of a species necessary, but to also try and do everything else regarding bringing back a species, whether it's putting an elephant egg into a lion and producing a lion, or so reproductive methods, all these methods that you can do to save a species. You really need the genetic material to do anything. So I think that's probably the most important thing. I'm very interested to hear that you lot at the Ark now have got interested in the invertebrates, because I think that, and we can do our English ones with ease, particularly in this time when no one can really collect much or go and do much. But I think it's absolutely vital, not only for the animal and the biodiversity on Earth, but also for us. I mean, the insects and things are the ones that fertilize all our crops. I think this hadn't been grasped, and it's for your generation to, stand and get something going on this.
So do you feel like going into the preservation of insects and things like that, that's a direction you'd like to see the Ark go in the future, to sort of focus on that side of things?
Well, I think we can in England here. I mean, if you look at our invertebrate species, and there's one only has to go down the garden or drive one's car at night with moths and going on the windscreen. And that was the first time I noticed insects, because 20 years ago, night you had to have your wipers on and water pouring, because the insects were banging on the windscreen when you were driving in the dark, because they came to the light. And now, not one. I mean, that's just a simple little thing one noticed, that everyone will have noticed. And I think for one loves animals, and even if one didn't, I think one could convince people that it's for their own benefit. And I just see creeping in little stories that seem to be saying that, and I'm interested in you guys concentrating on the invertebrates in the UK.
Yeah, I think definitely if it's something that's as stark as that, to be noticing it, those sorts of changes, it's just terrifying that we need to be doing something about it, really. So also, could you tell us a bit more about the sort of work you were doing before you started with the Partulus snails and the Frozen Ark, and some of the other things you've been involved with? You said that you're sort of interested in immunology as well, and some of the stuff you've done there.
Yes, well, I did a PhD as a result of my work in Oxford. One of our colleagues was someone called David Kirby, who was interested in why mammalian mothers don't reject their babies, because they've inherited half their genes come from the father, and should be antigenic to the mother when she bears a child. And this was I liked the immunology course, and this was a very interesting problem, because it also has implications for grafts, grafting people with organs that they always reject them. So what's the mother doing? What's the mother's immune system doing to stop their rejecting their babies? Is just a natural thing. And how can we incorporate that for patients who have transplants and things? That was the question. So I did this. I got interested in this question of why we don't reject our offspring. And I was doing this in mice. And then Ann Clarke, who, when we were research students, Edinburgh was good because it gave you 4 years. So your last year was on research, and you chose your research. And I chose something that on these lines with mice. So I had inbred mice, fathers with different genetics to mothers. So then we were looking at the offspring. What was their genetics? And then we were immunizing the mothers against the fathers. So she had a good immune response to the father's genes, and then mating her. They were all inbred. So we knew we could look at 1 or 2 genes. Even when the mother was immunized to the father, she didn't reject her babies. But then, of course, there's lots of other pregnancy problems. This was thought rather a fascinating subject. And we did it in mice and got various papers out of it, and learned a few things and got some increase. When one gave 2 skid grafts, there was a reaction in the mothers, and they did reject their babies and they aborted in much higher degrees. So I did all that and I got my PhD. And then I was going to go on to do other things, and I got waylaid by brown snails. And so I went as part of the teams to collect the samples in trips to Polynesia. We made about 3 or 4 trips, and then I got hooked on the fact that these things were dying out. And I didn't pursue my immunology studies, but instead we set up the Frozen Ark project about 17 years ago. We set up the Frozen Ark in Nottingham University, and in the Department of Zoology, and we had the ground floor. And when you explore where the Ark office is, you'll see that was our space. And so Brian had a room, I had a room, and we started to set up the charity. So off we went there, really. And then it was 10 years of excitement. We got money. I got to travel all over, giving lectures the world, getting consortium members aboard. And I did that for the next, I suppose, 20 years, until I got to 80. And then I thought I must step down and leave you guys on to carry on. But I have promised to write this book, which is basically the story. And I think I must get back to that. Maybe I will when COVID's over and I can go back to college. So that's the sort of main sort of story, and it's still going on. I mean, the zoos are releasing them back into the wild. It's terrible that Trevor Coots has died of COVID, and I think we've got to wait for someone who's going to take his place in Maria. Just hope there's somebody in the art that might take that job on, because that's the end of the story. And all this business, how they're seeing how they live at the ones at high altitude. I think there's a rumor, I don't think it's fact yet, but the partridge that live at very high altitudes, and you can, tell me if this is wrong, but this was a story I heard some time ago, possibility that they're living at high altitudes, and the Ugandina, the predators doesn't like altitude, so it's not going up. So this is a story maybe of population, of extinction, and of repopulation if a species is able at high altitude in this case. So that's really the story.
Yeah, that would be really interesting if they have sort of been able to find something that they can escape from it, really.
Or it's just those that evolved to happen to like high altitude. Yeah. The benefits are not to be eaten.
And while you were doing your PhD, was Ann Clarke, she was your supervisor at the time, is that right?
Yes, she was. Yes, I was lucky enough. We had to give a small talk on our projects, and Ann Clarke came to listen to them as undergraduates. And she came up to me afterwards and said, "Would you like to do a PhD with me on this subject?" That was the mice. And I said, "Well, I'd love to." Terribly shocked, really. Someone should do that to me. But I said, "I'm terribly sorry, but I'm just off to Polynesia for 6 months." And she said, "Oh, well, when you come back, start." So I did. It was as easy as that in those days.
And also just wanted to ask if you sort of have any advice for students who are thinking of coming into a career in conservation and biobanking and things, if there's anything you'd suggest for people of sort of our age who are studying and looking at what to do next, really.
I think it's absolutely vital. And I think we haven't begun to address that problem, not only for the world. I mean, now we've got climate change, and no one knows quite what it's due to, but the story is it's due to rising sea levels caused by the melting Arctic. And the whole ecosystem. I mean, apart from the horror of it all, it may actually be a necessity for us to have wild animals in the world, because they've taken millions and millions of years to evolve. And the idea that one species can just go and wipe them all out to suit it at that particular moment. I mean, even to shoot them for fun and to be down to I mean, it's amazing that they're worried about the elephants now. But I think, I mean, one has to, you can also, I think what the Ark ought to do is to probably, to make things a little bit easier, ought to specialise, and we have to some extent, but not entirely, do eggs and sperm of all animals, endangered animals, not just cells. Because in the end of the day, you can probably get a sperm or an egg out of a cell if you do enough experiments, but you need the sperm and the eggs, so it's easier. So you can do, you can implant the fertilized egg into the remaining animal if there's no. I mean, there's the rhino. I think the one-horned rhino. I think there's only females left, and I think it's got to be a global thing, because I think the trouble about Britain is we haven't got. If you look at the percentage of species we have, endangered species we have, compared to somewhere like Africa, we haven't got much. We've got many more invertebrates, which are why I think it's a good thing for art to do easily in these circumstances. But this has got to be global. So although we were global before and we're not so, you're not so global now, I think we have to go back to global for it to be meaningful. So I think you should all fight for collecting the world's endangered species, not just English ones, because most of our vertebrates are in Europe anyway. But you need people spread around the world who've got collections. Now we have to some extent, but I think that part maybe needs invigorating and encouraging more of. They all need contacting, of course, the next generation down. They've got to be contacted, not just our generation's colleagues.
Yeah, definitely. I think nothing's going to happen if it's not a worldwide effort. We can't just work from one aspect.
You can develop techniques and you can start to show people how you do it. But then because most of the consortium members were working from the same batch of instructions to do it in this way. So there will be some conformity. And with invertebrates, it's much more difficult. You can't just take a cell or two. You've got to work it all out. What do you do with a fly? Do you freeze down the whole fly, or do you take some cells and make single cell samples and freeze those down? And what about sperm and eggs? Huge area.
And I think sort of the final question from me is just to say, obviously you've been like a really successful female scientist in your area, and lots of your work has been sort of inspirational to women around the world and things. So do you have any advice for sort of aspiring female scientists who are trying to get into the world of STEM and things like that?
I don't know, but I think you're all doing rather well, aren't you?
Yeah, placement students are doing well at the Frozen Ark, yeah.
Yeah, but all female undergraduates, I think, doing well. It was really, I was really lucky to have Ann Clarke as my supervisor, because she was an example of someone of her caliber, and she was a member of the Ark. Of course, she was on our board, and she was, she showed me how to do it. You just carry on, and It was quite a shock because just the ease, I'm sure it was, in a sense, there were many fewer of us, but it may be just because it was, I think I only did it. Well, my husband was always very helpful in me getting to university, because when we got into it, I thought I'll change from being a reporter, and then I did 3 years as a technician. And then I got interested in that subject and colleagues and things that interested me and various aspects of it. And I thought I've got to go to university, so I went to night school. I had zoology, I had botany, I needed chemistry. And so I had to go to evening classes. And then when I got to first year university, to get a science degree, you had to do physics. And I knew no physics at all. So my husband, they wouldn't give me a place in this high level physics. So my husband tutored me with the 5 questions. We looked at all the papers with the 5 questions he thought were going to come up in GCSEs, O level, it's called then. 5 weeks he looked up the questions, and he taught me 5 answers to 5 questions, and one of them came up. 4 of that, I think it was 3 out of 5 or something like that. But it did enough to get me through 5-week course. Did enough to get me through 3 of the questions. That's probably not good to say that, is it?
Well, it worked out okay. So
it worked out okay. So I think it's just, if you want to do it, you do it. Yeah, and there's no reason why, because we're female, we can't do anything, and we all know that. Quickly, if we've got a minute, tell me what you're all doing.
So we did some field work while we were back in Cardiff, and so for the part of the placement, I'm doing an investigation which is looking at different mammal survey techniques and comparing those. So that's my project for this year, and then be going back to finish the final year of my biology degree next year.
And do you have to do a project for that?
Yeah, for our final year we have to do a project as well, but we haven't got
what's going to be?
Mine, I'm not actually sure yet. We haven't got to the stage of choosing it just yet, but I'm hoping to do it on something sort of animal based or conservation based, if possible.
So I worked on the field work with Rhian, and what I'm doing is comparing sort of different green spaces around Cardiff Uni. So there's like sort of main building, they've got like a nice manicured lawn, and so we put some footprint tunnels there and found hedgehogs and mice. And then we were also looking at Butte Park, and so there was a lot more sort of diversity in Butte Park. And yes, I'm just, reading papers and looking at different ways to analyze the results. And then, same as Rhian, going back in the fall and picking a project, either conservation related or sort of more disease immunology based, or just sort of see which ones appeal to me most. And then, I'm not too sure after that.
Well, good luck.
Thank you.
So I've been working on looking into how biobanks can be more environmentally friendly. And then the goal is to make that into a guidance document that biobanks can use and hopefully reduce carbon emissions. But what we've all actually been doing as well is a lot of the public engagement stuff. And so we do a lot of social media posts, and I'm really interested in that. So I'm hoping for my project next year, some of our professors do public engagement, and one of them in particular wants to, if COVID allows, go into schools and develop some science communication materials for them. So I'm really hoping that I can do that.
Oh, that sounds great. I think my nicest audiences, without fail, the talks I gave to school children about the Ark. They were so enthusiastic. Mm-hmm. So I think there's hope for the future. And I don't know if that would have been true probably when I was young and someone came and told me things like that about the Ark, whether we would have been. But there was something that got them about it, that, academics were just merely some of them would look bored, academics who weren't in the field. But these school children were never, ever with one look bored. Yeah. So I have hopes of the next generation.
Yeah.
Well, 2 generations.
I think that's been one of the saddest things about doing our placement in COVID is we haven't really been able to have many face-to-face interactions.
Yes.
And I love kids, and I would really love to develop some science communication with them, because
yes,
kids are really enthusiastic about these things. And I think especially endangered species, a lot of my younger cousins are interested in it, and I can have conversations with it, and they talk about it in school. So I think there's a great opportunity to show them what we're doing and inspire the next generation to be
absolutely. I mean, that's what probably we failed at in a way, but I think there's a sort of, it's almost I've got grandchildren who are fascinated. So it's also skipped a generation, a bit maybe.
Yeah.
But now it's serious. I mean, it's not just interest now, it's survival. And you don't hear that talked about much. The consequences of loss of biodiversity to us is a subject I read very little about.
Yeah, exactly. I think we have a tendency, as a species to only think about ourselves, and we don't, I mean, I think it's a tragedy that we're losing so many species anyway. But I mean, we don't stop and think what that means for us. How can we live on a world where most of the biodiversity is gone? It's just...
Well, we can't. I mean, it's not just the fact that we won't be any beautiful butterflies to look at. It's the fact that what's going to fertilize all our crops. There are mid-trillion things that we don't yet know what they do because they haven't been studied enough. So I think that the new direction, although we've got to save the vertebrates, that's being more taken care of than the invertebrates, of which all the vertebrates depend also.
I've been working on the particular species in the Frozen Ark freezer at University of Nottingham. So all the snails, they are like in a box. So what I'm doing is to give each of them a label and put it on my system, like a database of the snails, so we can easily identify the species and match it with the location and other things that's attached to it. Then I've been working in the lab too, like I'm extracting some DNA samples from the snails, then and also dissecting them, because we're trying to know the genes that is necessary for the formation of maybe a dextra or sinistra chirality of the snails.
Yes.
So basically that's what I've been doing here, and also involved in public engagement as well.
Well, that's interesting. So who are you working with?
Angus Davies.
Angus. Oh, yes. Well, of course, I remember Angus very well. You must give him my best wishes. And he's one of the people I know that work harder than anybody else. You're a great man. You're lucky.
Yeah, so I've just been doing the prioritisation tool, which was started last year or the year before by Sarah. So it's just a tool to kind of narrow down what species need to be collected as soon as possible for the biobank, so that time isn't being wasted by thinking about who should be next or getting collections that aren't needed. So we're just being the most time efficient with our collections.
That's great. So what areas are you concentrating on? Any areas for which you're collecting, or?
Yeah, so it's so there's different weights of how. So it will be a tool that will be put onto a website. So just from coding and all that sort of stuff. And there's different weightings on demand or storage, collections, and their conservation value, so that other people could change their weightings to see how they would want to and what species they would want to collect, depending on other ones. So it'd been compared from like Frozen Ark to Cryo-Arks and other different biobanks, and see how what they specialise in and how the weightings change for everything.
So you're working with Sarah?
Yeah, so she's obviously started all the work, and then we're working together and with Math to try and help finish it all.
Oh, that's good. So where are you located? Are you in the Queen's Medical Centre or in zoology?
Well, because of COVID, I've just been working from home in Kent. So
Can you do practical work? No.
Well, I did. I went to the labs in Nottingham with Olu for a week so that we could sort out all of the samples in the freezer. so that they're all barcoded and we've got our complete database and everything. So I was able to do that a couple of weeks ago, really. So that was a really good experience.
So you feel that things are getting a bit better. You'll be able to do a bit of practical work sooner or later.
Yeah, definitely.
Oh, well, give my love to Nottingham.
Well, thank you so much, Anne, for talking to us and going through everything. I mean, it's just such an amazing story of the whole charity. So it's been lovely to talk to you.
Well, it's been a great pleasure for me. Good luck to everyone, and I look forward to hearing how you're doing.
Thanks so much, Anne.
Thank you.
Nice to see you.
Thank you.
That's the end of today's episode. We all really enjoyed chatting to Anne and hope that you enjoyed listening. That was the final episode of Series 1 of the Frozen Bytes podcast. I've really enjoyed being the host for this first series and have met some amazing guests. So it's goodbye for now, but we hope to release new episodes in the future.